Yes. Sure. You can put it in the beginning chat. Raise your hand if you want to get this. Can we get water up here? Yes.
Want one now?
No.
All right. Well, it's 5 after 5, and I think we'll begin. Welcome to the 24th annual Paul S. Russell Lecture in Transplantation. Dr. Russell, who turned 100 in January, sent his regrets for not being here, but he's joining us at home by Zoom, so he'll be participating. Before starting the program, I'd like to give out a few thanks. First of all, to Sanofi, Alexion, Takeda, and Lung Bioengineering for supporting this event. Thank you very much. Also to Tony Rogers and Laura Kupferberg, who organized the event. Tony and Laura, thank you. So, given the extraordinary breadth and impact of her accomplishments, introducing the 2025 Paul Russell Lecture is daunting. Finding words to describe Dr. Martine Rothblatt is difficult. Capturing someone whose contributions span not only aviation, satellite communications, biotechnology, cyber consciousness, and transgender rights is challenging. The best I and others could come up with were words like inventor, pioneering entrepreneur, visionary, polymath, and my favorite, futurist. Still, none of them capture the person who, with a background in business, law, and ethics, has reshaped multiple industries and pushed forward the boundaries of science and technology. Dr. Rothblatt first made her mark in founding Cirrus Satellite Radio. This transformed how the world listened to audio content, but her most profound and personal impact came in medicine. When her daughter was diagnosed with life-threatening pulmonary hypertension, Dr. Rothblatt formed United Therapeutics, a biotech company focused on therapies for rare diseases. There, she discovered drugs that not only saved her daughter's lives, but saved the lives of thousands of others with pulmonary hypertension. Under her leadership, United Therapeutics has gone on to become a trailblazer in regenerative medicine, organ bioengineering, and xenotransplantation. As I count them, Dr. Rothblatt can claim at least 4 firsts in the field of transplantation, but I'm not going to steal her thunder. I'll let her tell you about them. Dr. Rothblatt has received numerous awards and honors, but the one most impressive to me is the Benjamin Franklin Medal for Distinguished Achievement in Sciences. This accolade places her among the ranks of Linus Pauling, Watson and Crick, and fittingly, Tom Starzl, who, legend has it, was the first to convince her that xenotransplantation would become a reality. Beyond her scientific and entrepreneurial achievements, Dr. Rothblatt is a thought leader in the ethics and future of cognitive software. She has written extensively about cyber consciousness and digital immortality. She also is a powerful advocate for transgender rights, diversity, and inclusion. For all these reasons and more, it is my honor and privilege to introduce to you Dr. Martine Rothblatt, the 2025 Paul S. Russell Visiting Professor.
That is such a beautiful introduction. Thank you so much. I've never heard one better than that, so I will do my best to live up to it. I think my first step in living up to it is maybe finding the mouse. Okay, I've done that. It's an amazing honor for me to be here today. First and foremost, I am especially humbled and honored to be in the presence of Paul Russell. And I thank you, Dr. Russell, via Zoom, and happy birthday on your 100th birthday. Second, MassGen is such a legendary organization and hospital. It features larger than any other medical establishment in my mind, in terms of a place that is literally a citadel of blazing the way forward in medical research and medical technology. So to be here and to be an invited lecturer and to be given such a kind introduction, um, I really am pinching myself. It's unbelievable. Third, I'd like to say a humongous thank you. I have so many people in the audience here who are like the milestones of my life. And the very first time I came to MassGen, it was to meet and be educated on thymic tolerance by David Sachs. And then to see David Sachs here in the audience today, it's like, wow. And the greatest educations I had on transplant immunology were by way back working on my Ph.D. in the 1990s, reading even by that time the voluminous publications of David Cooper. And David Sachs was also had voluminous publications at that point in time. And then here is David Cooper in the audience. As mentioned, our daughter has pulmonary arterial hypertension, and thank goodness from our development of multiple medicines, we have 4 FDA-approved medicines for the condition. She has not yet needed a transplant, which is still the only definitive cure for pulmonary hypertension. But in trying to figure out what kind of medicine could be used for this mysterious disease, way back in the early 1990s, where I jumped from Sirius XM to pulmonary hypertension medicine was with the Fishman on pulmonary disorders, the illustrious father of Jay Fishman here. And so to have Jay like front and center with me, I'm extremely, extremely humbled. And then over the years, as we began to develop the concept of xenotransplantation for the lung, which was really the last organ anybody was interested in for xenotransplantation, I find out that sort of a stone's throw from our home in Silver Spring, Maryland, is the world's #1 expert in xenolung transplantation, Robin Pearson. And here is Robin Pearson in the audience. So I could go on, but I guess I'll use up all my lecture time. It's just an amazing, amazing moment in my life. And thank you so much for making this happen. All righty. So the title of this presentation is The Future of Organ Transplantation. Actually, the organizers gave me no clue what to talk about. They said I could talk about anything I wanted to. I am not going to talk about things that are like the past of organ transplantation, let's say routine allotransplantation, even though, you know, the first allotransplants between, you know, identical twins were done right here in this ecosystem, because all of you are quite expert in allotransplantation, and there still is a lot to learn and a lot to do, but I don't think I would be really the right person to talk about something that was decades past. And all of you are steeped in that every day of the year. I'm also not going to talk today about xenotransplantation, because contrary to whoever was the villain, and different fingers are pointed in that direction, who said xenotransplantation will always be the future of transplantation, I don't think that's any more the case. I think xenotransplantation is now the present of transplantation. And we have 2 lovely individuals walking around pretty darn healthy for their pre-existing conditions with xenografts, one about 2 months out and one about 4 months out. And there were patients before them. And I would say there is like zero doubt that there will be dozens of patients after them within the next couple of years. And there is really no real doubt that there will be hundreds of patients after them in the next 5 years, and then thousands. And we get to the kind of numbers that you see with allotransplantation today. So I would never be a person who would say that xeno is the future of transplantation. I hate when people say that, actually. I say it's a present of transplantation. But I will talk about something that I do believe is the future of transplantation, and not in the sort of disparaging way that people referred to xenotransplantation as the future. I think in a way that shows that the pieces are rapidly falling into place. And those people in the audience that would want to have, I think, the biggest impact on the future of organ transplantation, I would encourage you to pursue the type of directions that I'll describe in this lecture. And I can say from my own firsthand experience, I spend most of my time obsessed on the vision that I'll share today for what I consider to be the future of organ transplantation. I reserve a little bit of time for the aviation and the cognitive consciousness, but I need that to help me with the future of transplantation. All righty here. So most future organ transplants, I believe, will be lab manufactured in quantity. This can eliminate many of the ethical problems in allotransplantation today. However, there will surely be an important role in the future for allotransplants and xenotransplants, as well as many biomechanical organs to play for many decades to come. I'm reminded of the saying from the famous futurist, also a fellow radio engineer and a science fiction writer, Arthur C. Clarke, that as no technology ever completely disappears, but just becomes less and less important as the technological horizon widens. So I think all of the kinds of transplants we're doing today will continue to be done, but the kind of gold standard will become lab manufactured organs, which is something that is not really done right now. Lab manufactured organ alternatives are also called GMP organs because they are manufactured under the FDA's required good manufacturing practices, or GMP. Making GMP organs follows a system similar to that of making a garden. First, you shape the garden. Second, you prepare the soil. Third, you choose the seeds. Fourth, you sprout the seeds. Fifth, plant the seedlings. Sixth, tend the garden. Seventh, harvest the garden. And eighth, eat the harvest. For GMP organs, we first shape the organ alternative with a scaffold. We prepare the scaffold with peptides that enable cell adhesion and cell growth. Third, we choose the cells that we want to plant in the scaffold. Fourth, we expand the cells like sprouting seeds into seedlings. Fifth, we cellularize the scaffold with the expanded cells. Sixth, we nurture the cells into homeostatically functioning tissues. Seventh, we harvest the matured organ alternative for transplant. And finally, eighth, we expect the body to integrate, or that's the way the body would eat it, to integrate the GMP organ via cell migration, scaffold absorption, and physiologic regeneration. To shape an organ alternative, it really means, in terms of the garden analogy, to create an organ scaffold. Organ alternatives can be algorithmically shaped for the desired functional equivalence to native anatomy. To be clear, these abstracted organ shapes are going to diverge from an idealized anatomic organ. But my hypothesis is that the difference won't materially matter. Sure, the shapes of our anatomic organs were beautifully hued by eons of evolution, but that doesn't mean an abstracted algorithmic model couldn't function at least nearly as well. After all, we make these kind of substitutions with heart valves, contact lenses, and many other types of medical devices. As a pilot, I can't do a slideshow without one airplane picture. In a similar vein, one does not need to build a bird to fly just build a plane that performs the flying functions of a bird. The zero carbon footprint organ delivery aircraft shown here in juxtaposition with the Arctic tern was modeled after that exact bird to achieve a 250 nautical mile organ delivery range with just rechargeable batteries and a zero carbon footprint. Yes, we just needed the range of the bird, and hence its very high lift to drag ratio. We did not have to replicate the digestive, social, or reproductive functions of the bird. Even the smallest bird is comprised of the smallest bird's comprised of billions and billions of cells, but even the largest airplanes have only a few million parts, and many much less than that. I think my little soundbite here would be that biomimicry of function or biomimicry of function for anatomy does not require replication of the structure of that anatomy. For the kidney, to shape the garden means to provide pathways for creatinine clearance and urine production. To achieve clinically relevant scale, about what we do at United Therapeutics and under the leadership of one of your alumni, Herodot, over in Medford across town, to achieve clinically relevant scale, we use 50 tubes that are placed in a single layer, providing a cumulative surface area of about 10 to 20 square centimeters. A single layer can accommodate a blood flow of approximately 10 milliliters per minute and clear up to about 1 milliliter per minute. We achieve therapeutic power by then layering sheets vertically, one over another, with a vascular distribution system that allows surgical connection to the patient's vascular system, such as the iliac veins and arteries. Due to low resistance, there's no need for mechanical pumps. The blood flows naturally through the organ alternative. The entire surface area is then lined by endothelial cells to avoid formation of blood clots, while the tubes within the sheets are also lined with podocytes to perform filtration like the kidney's glomerulus, and tubular epithelial cells for reabsorption like in a subsequent section. The total number of layers is 32 layers, and it ends up providing the total amount of vascular clearance equivalent to something close to half of a native kidney and about 4 times better than is achieved with dialysis.
Lung biotechnology is developing an unlimited supply of patient-specific lungs. In partnership with 3D Systems, Lung biotechnology found new purpose for the digital light projection chip, used for years to bring film to life with pixels. Where others saw car chases and sweeping landscapes, Lung biotechnology saw a breakthrough, to print vasculature in native fidelity. Using the latest hydrogel biomaterials, these pixels can create organ scaffolds at subcellular resolution, sustaining the over 200 million alveoli and 8 billion cells of each lung. Inspired by nature, these scaffolds mimic lung extracellular matrix and provide oxygen through capillaries to support cells.
As mentioned in the preceding video, a crucial material called hydrogel is used for both shaping the scaffold and for its subsequent cellularization and integration into the body. Hydrogels are water-based materials with embedded polymers that can be cross-linked by the lasers you saw in the video to provide a very highly defined structure and very highly specified physical attributes. Hydrogels were first identified in 1954 and found their first mass biomedical use as soft contact lenses in the 1970s. Only in the 21st century, though, our time right now, did we see a new wave of hydrogel physical-chemical methods and structural modifications to improve the mechanical properties of hydrogels by the incorporation of different cross-linkers, changing the physical architecture of polymer networks and utilizing natural synthetic polymers just within the past 5 years have allowed us to shape the scaffold so precisely that we can achieve the type of dimensionality that you see in an anatomic organ. That video, this next video is going to show off some of the mechanical properties of these hydrogels.
Okay, I crumple it. Now hold it up and show that it's still like the same shape.
Yeah.
Wow. Yeah.
Okay, you just squeeze the hell out of it.
I believe hydrogels will be a defining material of the 21st century, just like plastics were a defining material of the 20th century. That belief inspired me to add a little bit of AI to the following video, but I wonder how many of you can remember the punchline of this next video. There you go, the winner. Let's see how I get this video to play here. Plastics, everybody is winning the prize. But we got to hear it.
Just one word.
Yes, sir.
Are you listening?
Yes, I am.
Hydrogels.
Exactly how do you mean?
There's a great future in hydrogels. Think about it. Will you think about it?
Yes, I will.
Enough said. That's a deal.
I guess that movie made a lasting impression on me. I think they were prescient to predict plastics would become such a big thing, maybe too big of a big thing, back in the 1960s. But now I think that the key material for the future of transplantable organs is hydrogels. PubMed citations of hydrogels have increased exponentially, and when the purpose is to be used in a 3D printer for biology, then the hydrogel becomes known as a bioink. There are an infinite number of possible hydrogel bioinks, and it is important to chemically design a hydrogel for its intended purpose. We have made, within United Therapeutics, hundreds of novel hydrogel bioinks to get the ones that we like best. For example, it must make a lung or a kidney flexible enough to be, in the case of the lung, moved by the diaphragm, which means a modulus of under around 100 kilopascals, and yet firm enough to make it through a multi-week-long 3D printing process with the kind of millijoules of energy that these lasers deliver, without, in the end, any damage, such as either swelling on the one hand or tearing on the other hand. Finally, hydrogels must be surface modified for success, because ordinarily cells will not lay down on them just as they are. Now that is where we have shaped the garden with a hydrogel scaffold, but it's now time to fertilize the garden, fertilize the soil, maybe pick up some organic soil, sorry. In other words, it is time to prepare the garden that we have shaped. That's what we call peptide surface modification. Once a scaffold is printed, its inner surface needs to be sprinkled with specific peptides that are known to have high affinity for specific cellular membrane-bound molecules, such as integrins and syntechins. From trial and error, we have learned to use a peptide cocktail. Specifically chosen peptides seeded at high density give the membrane-bound integrins on cells a handhold, triggering the creation of intracellular focal adhesion complexes and the secretion of extracellular matrix. The peptide cell interaction ultimately enables intercellular signaling that then leads to tissue formation. In this image, the green, red, and purple or magenta squiggles represent peptides that we have scaffold fertilized. The technical word again is modified, specific to the integrin, syntechin, and ECM binding domains on the cells. We coat the scaffolds with these peptides after they are shaped. The gray puzzle pieces in the image toward the bottom are the cross-linked PEG diacrylate molecules that make up the hydrogel scaffold structure. The top half of the picture shows a curved section of a cell's membrane becoming seated atop peptides, that's being seated atop peptides. The arrow to the right indicates that some of these peptides are for binding to an extracellular matrix that cells start to produce, but only after the integrins and the syntechin binding has been achieved. This image pans out from the one that I showed just before and indicates some of the many changes that occur within a cell once it binds to the peptide surface modified scaffold. Signals for growth and other functions are sent inward toward the nucleus, and even the cell's cytoskeleton actually begins to change shape. Just like a vegetable or flower garden, seeds will do very, very different things if they are put on dry, hard dirt compared to those that are placed within moist, richly endowed soil. Also in a vegetable garden, there is much more volume of fertilizer than there is volume of seeds. And so it is with peptide surface modification. We surface modify at a density of somewhat less than 1 micromole per square centimeter, which implies billions of peptides per cell. This is interesting because integrin numbers per cell are much, much lower, only a few hundred or thousands per cell. But based on measured binding kinetics, our computerized digital lung twin team predicts that peptides need to outnumber integrins by quite a large number in order for successful integrin saturation to occur, as it does in our 3D printed lungs. The last part of the soil preparation process in our garden for the GMP organs are to wash the scaffolds of unreacted hydrogel material from printing, to rinse the scaffolds after surface modification, to remove the large number of unreacted to remove the large number of peptides after surface modification that did not participate in cell adhesion, and finally to precondition the scaffold with media especially conducive to the type of cells that we want to grow on that scaffold. Choosing the seeds. You see, already people say, oh my God, Martine, this is a hell of a lot of work to make this organ. And we thought it was a lot of work making the xenografts. I said, no, the xenografts are easy by comparison because the pigs do all the work. We have to do the work. We technicians and scientists have to do the work with these. So now that the garden is shaped and the soil has been made seeding friendly, we're ready to choose the cells that we want to grow on the scaffold. As to the types of cells that we introduce into the scaffold for peptide enabled colonization, there are broadly two choices: allogeneic master cell banks or autologous cells derived from the intended organ recipient's own DNA. With allogeneic master cell banks, or MCBs, cells are removed from an organ of an organ donor when the organs could not be used for transplantation. They are then carefully screened to ensure their absolute health and safety. They are then separated by cell type and expanded several billion-fold. In this way, one organ donor can provide the cells to create dozens or even hundreds of GMP manufactured organs. I mean, that's a pretty beautiful thing. Somebody wanted to be an organ donor, their organs couldn't be used immediately in a person, but because of that organs they donate, there ends up being hundreds of organs able to save lives. So it can do a lot more good than one would have thought. We actually use multiple donors so that we have master cell banks for different ABO and HLA groups. With autologous cells, the second kind of pathway or second broad type of seed to plant in these scaffolds, the process begins by separating special cells from a patient, such as CD34 or PBMCs, that can be reprogramming using Yamanaka factors or newer RNA methods. After that, the resulting inducible pluripotent stem cells, or iPSCs, can be further differentiated into the types of cells desired for cellularization. For example, podocytes for the lab-produced kidney, or airway cells for the lab-produced lung. As shown in the center of this slide, it is also possible to precede such differentiation with genetic engineering to avoid using cells with known disease-causing polymorphisms. Many billions of such cells can be made in laboratories today. At our company, United Therapeutics, between our kidney cells, our autologous cells, and our master cell banks of allogeneic cells, we make over 2 trillion cells a year. Expanding, differentiating the scaffolds and cells. Having chosen the type of cells that we want to plant in the organ alternative scaffold, the next task is to sprout the cells, which means to expand them to the 5 to 10 billion cells each adult size organ requires. There are several well-practiced protocols for expanding different types of cells, and new protocols are being developed all the time. Most of our cell expansion occurs in 10-liter bioreactors, such as the one shown in this slide. The process is automated. We start by seeding the bioreactor with stem cells isolated from 2D flasks, which quickly clump together because they don't like to float alone. We achieve differentiation into different cell types by changing the media of the bioreactors, letting the clumps float to the bottom and periodically stirring the bioreactor to maintain good oxygen and pH. I'll show you a little video here in a second. As you will see in this video, and look to the bottom, you can see billions of differentiated endothelial and stromal cells. You have to take my word for that, falling in the clumps of thousands of cells per clump in the bioreactor. This kind of makes me think of an immense but microscopic snowstorm, but of differentiated cells instead of snowflakes. Although each clump of cells is actually as unique as each snowflake is. Yeah, you see all the snowflakes? These are all differentiated cells from a person's own reprogrammed CD34 cell. While it takes about 3 months for us to reprogram a patient's cells into an expanded population of iPSC cells clear of reprogramming tools such as Sendai, it takes only a few days for those iPSC cells to differentiate into very, very large numbers of endothelial and stromal cells. Similar methods apply to other cell types. Here's a close-up of clumps of the induced endothelial cells on day 4 of differentiation. I think this takes the concept of sprouting seeds to a whole new level. Now that we have plenty of sprouted seeds to plant, or we can say differentiated and expanded cells to place in the scaffold, the next task is to actually do the cellularization process, to actually plant the seeds. The cellularization process consists of injecting cells into arteries, veins, and/or airways, as is appropriate, of a well-prepared scaffold. The cellularization process takes 2 to 3 weeks for a lung and a little bit less time for our kidneys, and requires in each instance billions of cells. Here you can see our automated scaffold cellularization production line in Research Triangle Park, North Carolina. There are a variety of knobs on at our disposal to control the effective cellularization of a scaffold. These include cell type and cell sequence of delivery, as well as number and concentration of cells that are delivered, including precise control over pressure and flow rates during cell delivery to optimize the distribution of the cells across the entirety of the scaffold, from the proximal to the distal regions. Other knobs that we use on these machines, these bioreactors you see in front of you, include culture duration, application of physiological stimulation during culture, such as pulsatile vascular flow. Finally, of critical importance to the cell proliferation process is the media that we choose to use. And we constantly experiment with different medias, tweaking it all the time to achieve ideal and positively ascertained phenotype and function of our cells after they go through the kind of traumatic journey, a kind of a fantastic voyage for a cell through all of these artificial channels. The composition and timing and delivery of specific media components are precisely designed to optimize cell coverage and functionality. For example, we found VEGF very helpful in our media. As every gardener knows, how many of you are gardeners? Raise your hand. Wow, quite a few. No wonder you guys are such great biologists. As every gardener knows, it is not enough just to plant the seeds. Constant management is necessary to prevent overgrowth or underwatering, as well as insect control, just to name a few aspects of tending to a garden. Just as many responsibilities are required to tend a cellularized scaffold in a bioreactor. Attention must be paid on a daily basis to every metabolic detail to ensure cells are growing into healthy tissues and that the different types of tissues are all working together in harmony. Between exosomes and paracrine effects, cells are blabbermouths about how they are doing and what help they may need. And all of this has to be attended to in making a good and healthy garden. Managing all of the knobs for organ cellularization and maturation is as complex a problem as really solving multiple intersecting Rubik's cubes. Every face of the cube must be set correctly, and not only with respect to itself, but with respect to every other face of the cube. All of this must be done really upon largely non-invasive measures of what the cells are actually doing inside, what the tissue is actually doing inside the scaffold, and really the whole organ health. This is tremendously challenging, but we take inspiration from the comparably complex biotechnological challenges that have been mastered in our field already. And by our field, I mean our health sciences field. It's amazing what we do. All the keys we develop to unlock all the biochemical cellular pathways. Here you see a visualization of a portion of one of our 2,000 lobules that make up the 9 segments of the left human lung. We're able to use these type of digital twinning computer runs to save us a lot of time on balancing all the different faces of the Rubik's Cube. So we are running our cellularization process digitally in our computational biology lab. We call it Computational Laboratory for In Silico Molecular Biology, or CLIMB. And it literally lets us climb through the trial and error process of this very challenging cellularization much faster than otherwise, because we can visualize everything at a higher level in the in silico lung. It's sort of like, I think the saying that, I think Einstein said, no problem can be solved at the same level it was created. You have to rise up to a higher level to solve the problem. And the computational biology techniques and the digital twinning of our lung is, I believe, the secret sauce, if you will, that allows us to achieve a successful cellularization. This, by the way, the digital twinning, it also, not only does it save us a lot of time, it allows us to optimize exactly how much peptide needs to be deposited into the scaffold, which saves us millions of dollars on peptide. And it also saves us years and years of trial and error, because we can trial and error things 1,000 times faster in the computer than we can in the wet laboratory. But it is an iterative process, because all the data from our hands-on wet laboratory work, as shared with the computational laboratory team from RNA-seq data on the cells that we extract from the bioreactor, they then feed back in to their computational biology laboratory. So it's a synergistic process between the, if you will, the wet and the dry biology. The litmus test for GMP organs to be the future of organ transplantation is for them to work when outside of a bioreactor. This can only be demonstrated by harvesting the manufactured organ and testing it out. Here are some images of one of our lab manufactured lung alternatives that was transplanted into a pig whose gene that causes hyperacute rejection was knocked out. At the top left is the lung we manufactured after cellularization was done in the bioreactor, as I just got done explaining, but before transplant. In other words, upon harvesting from the bioreactor. On the right is a fluoroscopic video showing the injection of contrast into the transplanted left lung. You can see the contrast being injected into the left pulmonary artery and then spread through the tissue. The center bottom image is from a CT scan showing that the lung is inflated with air, which is dark colored, and is filling the chest cavity. These images demonstrate that manufactured lungs from the very same bioreactors I showed in the earlier images can in fact support life in a large mammal model, in this case, the pig. We now do these transplants we do more than one a week. And right now we are at several hours survival in the pig before the lung begins to fail. But we are on a path that I think by the end of this year will result in supporting the pig's survival for several days. And I think that will allow us then to take these organs to the next step, which would be testing them in a brain-dead cadaveric human model, decedent model, where we have succeeded in developing that model and tested kidneys for several weeks, in fact, up to 2 months. So we needed to develop that model for some of our xeno work, but I think that model will find its best use in these allogeneically and autologously cellularized lab manufactured organs. I believe the next step in manufactured organ harvesting and testing is to test them under ex vivo conditions in patients with acute organ failure for whom there are no other options but for whom the manufactured organ can be a bridge to recovery. Earlier this year, the FDA approved exactly this for the first clinical trial of our Miromatrix ELAP, or ex vivo liver assistance product. The clinical trial uses a lab-manufactured liver to help patients in acute liver failure live long enough for their liver to recover. And here you see in the upper left our liver manufacturing facility over in Eden Prairie, Minnesota, and a diagram of the protocol. This was the first FDA-approved manufactured organ study. It's in phase 1. And then a couple months ago, the FDA approved our Xeno kidney clinical trial. So I think the regulatory environment is actually very supportive of what's going on now in this field. For longer term post-harvest testing, the ideal preclinical model is the brain-dead, heart-beating human donor decedent model, also named the Parsons model by Dr. Jamie Locke, who performed the first such test with a clinically relevant organ. The ethics of doing this model have been accepted by a broad-based medical ethics working group led by Brendan Parent at NYU. The utility of this model was, I think, most clearly demonstrated when Dr. Robert Montgomery of NYU Langone, pictured here on the left, maintained the model earlier this year with nothing other than conventional immunosuppression treatment and without incidents of rejection that couldn't be handled with the conventional immunosuppressants and resolved. And I think, and as I just mentioned, running out to 2 months. Our anatomic organ never stopped changing. I mean, your organs are always works in motion and are more or less always in a state of regeneration and things are going good. Cells circulate throughout the body, following exosome, paracrine, and other biochemical signals to go to the places where they are needed. Allotransplants cannot reconnect every nerve, lymphatic drainage, and blood vessel as they were before the explant. Yet, when allotransplants are examined years later, it is amazing to see the extent to which new neural, lymphatic, and vascular connections regenerated and integrated with the allotransplant. It is a very big hypothesis of manufactured organ alternatives like the ones I presented today, that at least some useful organ physiology which is not provided will arise over time as endogenous cell migration and tissue growth occurs. The proof of that hypothesis will really take many years of post-regulatory approval experience in patients, as it has with allotransplants. In the meantime, if a manufactured organ alternative can provide enough organ functionality, perhaps in conjunction with pharmaceuticals, to give patients a good quality of life, that should be good enough from an ethical standpoint. I do believe that lab-manufactured organs are the future of transplantation. They have so many attractive features that it seems improbable for the technology to not be used in at least a majority of transplants, as those which such as those where the patient's health status can wait for several months, if not up to a year. Of course, allotransplants, mechanical organ replaced devices, and xenotransplants will also be used in thousands of patients annually. But as I describe in my book, Your Life or Mine, the latent demand for organ transplants is at least 10 times larger than the extant demand. When you hear there are so many people on the waiting list, the actual need is 10 times larger. I'd like to thank the many contributors and colleagues, team members that I have the immense honor to work with at United Therapeutics for everything they do day in, day out, and all the teams that they work with to provide the data, the possibility, and the hope that I had the privilege to share with you today. Thank you very much.
very much. Okay. Russell has a question.
No, Dr. Russell hasn't offered a question yet, but he wanted me to make sure that you knew he was watching and he appreciated your comments. So,
Thank you so much. Thank you.
Questions from the audience? Is it on now?
Thank you very much, Martine. That was a very stimulating lecture. I'm absolutely quite amazed at what you've been able to achieve so far. I had no idea that you were progressing so well. I really wanted to thank you for the contribution you made to xenotransplantation, because without you, I think the field would have collapsed about 10 or 15 years ago. You saved Revivicor, which was essential at that time. But going back to what you've told us today, I have a couple of simple questions. The first one is, when you showed us the artificial kidney that would be implantable, do you have to anticoagulate the blood for that?
Yes.
You do. So the patient would be anticoagulated permanently.
So I don't know that. I don't know the full answer to that question. I would say, you know, you're creating a very artificial environment, and cells react to artificial environments strangely. We go to the effort to line the all these tubes, these 50 micron wide tubes, stack after stack with endothelial cells, sort of with the hope that we would not have to anticoagulate. But nevertheless, I expect, especially in the beginning, that we will. Now, we'll find out very quickly, David, because that particular product has an amazing clinical development path that I think will move it much faster than people anticipate. So the clinical development path for that product, and again, the leader of that effort is a Mass General, Harold Ott, and I'm sure he would be very pleased to share the beautiful laboratory over in Bedford with anybody who wanted. But the scenario is the following. And we have 3 centers, and I would love for Mass Gen to join this idea that have already agreed to participate. A patient who is right now on long-term dialysis provides a cell sample from which we can create reprogrammed iPSC cells. Over a period of, roughly speaking, about, I would say, 4-ish months, we then expand their cells, differentiate their cells, cellularize the scaffold, and make for them in the bioreactor the kidney. Probably anticoagulated, just to be on the safe side. Then we go back to the patient, whether they're a Mass Gen patient or a Mayo patient or NYU patient, wherever. And the patient will have obviously consented to all of this when they gave the blood sample in the first place. And you could start off with like, okay, for this 4-hour dialysis session, 1 hour is going to be actually hooked up ex vivo to your own kidney cells outside of your body through this IVIVA kidney. And it should actually clear their blood better than dialysis, according to the stats that I showed there. There's no harm. It's only their own cells being given back to them. So no immunological harm at all. And then it's such a nice, I get excited about things that can be practically, clinically developed, because then you can go from like an hour to 2 hours to 4 hours. If we need additional ones, it's actually trivial. If you're going to make one of these kidneys, might as well make 10 of them at the same time. Okay, there's not a real limit on how many you can make. Certainly going to keep the patient's blood almost forever, as long as, again, they consent to all that. So I think with that type of clinical ramp up, we could very quickly get to the point where we can find out exactly how much, if any, coagulation actually is really, really needed. Okay. And then, hopefully as little as possible, and then be able to go ahead and get FDA approval to implant the kidney. And the patient is at that point free of dialysis entirely and feeling very much better. Should over time the kidney coagulate, or in otherwise dysfunction, You don't want to obviously do an operation more than necessary, but I think if the worst case, it kind of coagulated and you couldn't clear it every couple years. If I was on dialysis, I would rather have one of these Iviva kidneys replaced to me every 2 years than sit on a dialysis machine 3 days a week.
And the last question I have is the data you showed us with the pig lung. Is that an autologous lung? I mean, are the cells in autologous, or are they just identical pig tissues, or what?
So all these animals are getting, humans and pigs are getting a little bit mixed up here with the D cell and the recell and this and that. But strictly speaking, we would look at it as a human lung, albeit that particular one is on a D cell porcine scaffold. And that particular one is cellularized with a allogeneic set of cells from the master cell bank that I related to. I think that we could actually achieve that just as well right now today with autologous cells, because we make all of those four same types of cells. But as a practical matter, and a lot of times like laboratory work is just a practical thing, the autologous team is in one location, Manchester, New Hampshire, and the allogeneic team is in a different location in Research Triangle Park, and they each want to kind of do their own thing.
Thank you.
You may have already answered my question, Martine. That is this going to be individualized therapy? Every organ will be manufactured with cells from a particular recipient that you're going to be treating, as it was in the answer to that question, which is kind of enormous, complicated, but it would be autologous essentially for every individual to be treated. If not, I wondered why you have less or no immunosuppression, since any allogeneic or xenogeneic will still need immunosuppression.
So the autologous should not require any immunosuppression.
Right.
And that's what this is. And so that's what that refers to. The HLA matched allogeneic, I think, would require less immunosuppression than the xeno and the non-matched. So that's the less or the no. So I think that there are even better ways to manufacture organs with kind of like a universal donor cell, that you would not have to have like one organ, one patient. There will be many patients that they can't wait a year to get their organ, so they're not going to be satisfied this way. But for the patient who would rather not be on immunosuppression or could not tolerate immunosuppression and therefore cannot get a transplanted at all, I think this would be preferable. I think I tend to always just put myself in a patient's footsteps. And in this case, my continuing motivation for all of this is our youngest daughter. And I think, like, knowing her, she would rather not be on immunosuppression than be on it. I would rather her not be on immunosuppression than be on it. So I think that there will be a majority of people like that, but there will be almost as many people continuing to need allografts, xenografts, and new types of universal donor organs.
Well, for xeno, I think you'd be competing with the pig, but for allo, I agree with you. And I guess you could take advantage of linkage disequilibrium to set up one particular tissues that would satisfy a large number of individuals. It's still, individualized therapy is frightfully expensive.
It's an interesting question about the expense. You saw at the beginning of the slide that our company is a public benefit company. So we don't really let that factor into our consideration. And we just keep trying to develop the best therapeutic options for the patients. The expenses is such a strange question, because I read, like in the New York Times, that there are a gene therapy product that costs like $2 million. And it's hard to really imagine that it actually costs $2 million to make that product. It's almost unfathomable. What did they spend all the money on? So the I think all of these type of organs, and including the autologous ones, would probably cost in the neighborhood of low hundreds of thousands of dollars each. Every time you get an allotransplant, the bills that roll into Medicare or whatever are going to be in the low hundreds of thousands of dollars each. So I don't think that they're any more expensive than the allografts that people are using today.
Very thought-provoking. I have kind of two brief questions. The first is, you said you make trillions of cells
Yeah.
every year. Do they have the propensity to mutate to cells you don't want, and how do you prevent that or deal with it? And the second question is, what causes failure currently at 2 hours of those organs?
Yeah, so I have not personally received any reports of mutation of the expanded allogeneic cell. So I don't know that they mutate. I would frankly be surprised with those kind of numbers that there weren't some mutations in them. I think the approach of the FDA is that you have to demonstrate to them that it's either a very low probability or B, not a physiologic consequence to the patient. That 1 out of a trillion cells, say, mutated. I mean, you'll need a trillion cells for one of these organs, but 1 out of, say, 5 billion or so cells mutated, it was of no physiological or clinical significance. But I don't know firsthand the answer to that part of your question. And then what has caused failure so far with the recellularized allogeneic lungs is is blood clot in the distal portions of the pulmonary vascular bed. But it's mostly a matter of actually managing the animal model and building on what David said about proper coagulation protocol. So I'm confident we'll figure that out. I remember when working with Robin and David that, you know, all of these xenografts, they failed within 2 hours, within 1 hour, you know, within a half hour. They look horrible. At least ours don't look as bad as the ones that we saw in the early days of the xeno. So I think with persistence and continued good scientific practice, we'll be able to push those out to like a reasonable period of time.
That note, I think we'll end the session. It
Thank
was
you.
Fantastic. Thank you all for coming.
My pleasure.
Great job.
Thank you so much for hosting me, and thank you for your amazing introduction.
My pleasure. We'll see you.
Yep, I'll be.